I am grateful for Otto's introduction. My last visit to your department was more than a decade ago when both Otto and I still had black hair. Time flies fast. It is a great honor visiting you again. In today's seminar, I would like to present modern CT theory in a heuristic manner, with the hope that the audience, especially students, will gain insights and be inspired. By modern CT theory, I mean analytic cone beam CT reconstruction methods that use sophisticated mathematics. The treatment is rather challenging to engineering and computer science students. In contrast, my presentation will be very heuristic and hopefully easy to understand, especially if you already have basic knowledge on CT reconstruction and if you are motivated to understand the topic clearly. I teach medical imaging courses at RPI every year. On the left, you see the excellent textbook by Paul, which serves as our standard reference. However, the field of medical imaging evolves rapidly with new techniques, particularly deep learning-based imaging methods, emerging almost every month. To educate the next generation effectively, I continuously incorporate new material each semester. On the right is our own textbook, which serves as an introduction to the field of deep learning-based imaging, also known as deep imaging or deep reconstruction or deep recon. As you know, the dominant CT scanning mode today is called cone beam spiral CT. What is cone beam spiral CT? A point X-ray source and a detector plate define a cone-shaped X-ray beam that passes through the patient to collect cone beam projection data. The X-ray cone rotates within a CT scanner while a patient is continuously translated through the donut-shaped gantry. As a result, we acquire a stream of cone beam X-ray data along a spiral trajectory with respect to the patient. Currently, there are approximately 300 million medical CT scans performed worldwide yearly, the majority of which use the cone beam spiral CT mode. Would you like to learn how cone beam CT reconstruction is performed? There are now many algorithms for cone beam spiral CT reconstruction. I proposed the first cone beam spiral CT algorithm in 1991, which is an approximate algorithm. Most remarkably, in 2002, a Russian mathematician, Professor Alexander Katsevich at University of Central Florida, formulated the first theoretically exact cone beam spiral CT algorithm. That is, it took our field about a decade advancing approximate reconstruction to exact reconstruction for cone beam spiral CT. In my medical imaging course, I want to explain to my students what the fundamental ideas are behind image reconstruction, especially cone beam spiral CT image reconstruction, which is the main part of modern CT theory. More specifically, how can you perform approximate cone beam spiral CT image reconstruction? How could you improve approximate reconstruction to be theoretically exact? Given the cone beam divergence and longitudinal data truncation, how could such exact reconstruction be even possible? The paper by Professor Katsevich is mathematically very dense. I remember I challenged my group whether or not any of the lab members can understand every formula in Professor Katsevich's paper within 1 week. Here I will present a series of heuristic ideas, and then the exact formula for cone beam spiral CT image reconstruction will become clear to you. Since our students here are mainly in the Department of Physics, and we all understand wave-particle duality, let us start with the duality consideration. Duality is not just in physics. In mathematics and signal processing, we have duality as well. First, we have a wave point of view on the left. Fourier analysis is nothing but decomposition of a function into waves. Any smooth function, such as the sequence of blocks in red, can be represented as superposition of many sinusoidal waves in blue. On the other hand, we can have a particle point of view on the right, where the red curve is expressed as a collection of many blue bars or blue impulses. These impulses are delta functions appropriately shifted and scaled. If you view an image as a collection of pixels or voxels, you use a particle point of view. In X-ray physics, we have the Beer's law describing X-ray attenuation. Here you have an incoming X-ray beam with intensity I in. It gets attenuated by a piece of homogeneous material of thickness delta x. The beam density becomes I out after attenuation. This incremental attention process is described by the top-line ordinary differential equation with the proportional coefficient μx, which is called linear attenuation coefficient. Solving this ODE, we immediately obtain Beer's law on the second line, where we assume monochromatic X-ray energy. Beer's law links the incoming and outcoming X-ray intensities with an exponential attenuation factor whose index is the line integral of the linear attenuation coefficient distribution along an X-ray path. Practically, the incoming and outcoming X-ray intensities can be measured. Hence, we can directly measure the line integral with an X-ray beam. After discretization and assuming that each pixel is uniform and characterized by its linear attenuation coefficient, the line integral becomes a ray sum, that is, a linear combination of pixel values with weighting factors being pixel contributions to the X-ray path. Whenever you send an X-ray beam, you measure a ray sum, which is a linear equation. If you want to reconstruct a 2x2 image, you need 4 independent linear equations to solve 4 unknowns. Naively, we can use 2 horizontal and 2 vertical X-ray beams. However, these 4 equations are not totally independent, but if you use an iterative algorithm to solve this system of equations, you have a chance to recover the underlying image well enough. This toy example shows how to use the algebraic approach for image reconstruction. As a general question, when you solve the system of ray-sum equations, are you sure that there are enough independent linear equations to uniquely determine all unknown pixel values? I often use this illustration to help convince my students that a unique solution exists for image reconstruction from enough projection data. Imagine that an image to be reconstructed is an onion. We begin by shooting peripheral X-rays such that each ray intersects only a single molecule or a single pixel. In this way, we can directly measure each peripheral pixel using Beer's law. Once we resolve the outermost shell of the onion, we proceed to the next layer, shooting X-rays that now intersect each pixel in the next shell of the onion to compute their values. Shell by shell or layer by layer, we continue finding more and more pixel values until the entire onion is reconstructed. In other words, we can indeed gather sufficient information to peel the onion completely and recover the entire image. Naturally, we can extend the 2D onion peeling scheme to the 3D counterpart. Instead of using line integrals, in the 3D case, we perform X-ray measurements using planar integrals. Once again, each small particle in the outermost shell can be directly measured through a planar X-ray, integral over the plane intersecting each particle or voxel. Then, proceeding shell by shell, we can reconstruct the entire 3D onion based on the same principle presented in the previous slide. For image reconstruction, we can also take the other perspective, wave point of view. In this context, we treat an image as a superposition of many waves. In the 2D case, we have many 2D sinusoidal waves. These waves can be easily extracted by performing the Fourier transform of the image to obtain its Fourier spectrum, as shown in the middle picture. Each point in the spectrum represents a 2D wave field, as shown on the right for the red point. The larger distance from the origin in the spectrum, the higher frequency of the wave. The direction from the origin to the point is the direction of the wave propagation as indicated by the green arrow. To reconstruct this image, all you need is to find its Fourier spectrum. That is, all the wave components of the underlying image. Then you can perform the inverse Fourier transform for image reconstruction. That is, you can add all these waves together to recover the image. How can we find the Fourier spectrum from X-ray measurement? The magic phrase is Fourier slice theorem, which is explained as follows. Let us shoot a parallel beam of X-rays through the green object on the left, lowercase fxy, and form a parallel beam projection profile p theta r for a given projection angle theta and a detector location variable r. Then let us perform a 1D Fourier transform of this parallel beam projection profile to obtain a 1D Fourier profile uppercase f theta w, shown as a radial line profile in the 2D Fourier space in the middle. This is the famous Fourier slice theorem. If you keep changing the theta angle from 0 to 180 degrees, This radial line will sweep over the entire 2D Fourier space, measuring the whole Fourier space to provide enough information for the inverse Fourier transform, which is image reconstruction. Now we ask why the Fourier slice theorem is correct. If you read a textbook, you will have about 2-page derivation. We don't need to go through the details. Just look at the right figure and think about shooting vertical X-rays. These X-rays will measure all the information of waves propagating along the horizontal direction, which is orthogonal to the X-ray beam direction. You see some vertical rays measuring peaks of horizontal waves, and some other rays measuring valleys of horizontal waves. Note that for X-ray beams not orthogonal to the horizontal waves, you don't measure any information carried by the horizontal waves. For example, the oblique purple X-ray beam measures both peaks and valleys so that positive and negative half cycles of horizontal waves are cancelled out, giving you zero projection results. That is, you obtain no information about horizontal waves. The parallel beam scanning is the first generation mode for computed tomography, or CT. CT is nothing but image reconstruction from X-ray projection data, in this case parallel beam projections, based on the Fourier slice theorem. In the parallel beam geometry, each projection profile gives you a radial line in the Fourier domain. Many parallel beam projections over a 180-degree angular range form a star pattern in the Fourier space. We can perform the inverse Fourier transform of these data along the radial lines in the polar coordinate system. This will immediately give us the well-known filtered back projection, or FBP, scheme for image reconstruction in the red box. That is, Connecticut is a Fourier imaging mode. The inverse Fourier transform involves the double integrals in the top formula. The inner integral is a filtering process with the kernel defined in the bottom formula. After the filtration, the outer integral is a back projection process so that we can bring filtered projections back along X-ray paths over the field of view. Now we understand the key idea of parallel beam filtered back projection method. In practice, you don't measure line integrals one by one. To be efficient, we often use a point X-ray source and an array of detector elements to measure a fan beam projection at a time. This allows you to measure several hundred X-ray paths each time. In terms of information content, fan beam geometry and parallel beam geometry are equivalent. The left figure shows this equivalency. For example, the red line is measured in the parallel beam shown as the set of parallel purple lines. Also, this red line is measured by the two fan beams in light purple shading. This means that a 360-degree fan beam scan is equivalent to collecting a 180-degree parallel beam scan twice. While parallel beam data are indexed by two variables, detector location r and projection angle theta, fan beam data are indexed by other two variables, detector location t and projection angle beta. The two sets of variables can be easily converted from one to the other. The relationship and key derivation details are on the right figure, but don't worry about the details and just follow the general idea. After we have the conversion formulas between parallel beam variables and fan beam counterparts, we can immediately derive the fan beam formula from the parallel beam filtered back projection formula. This is easy. The red box contains the parallel beam formulas. Then we just need to replace the parallel beam variables with the fan beam counterparts and insert the 2D Jacobian transform. Note that the fan beam formula is also in the filtered back projection format, but with weighting factors involved. The derivation is kind of tedious. Let me make it heuristic here. Let us compare the parallel beam formula in the red box and the fan beam formula in the green box. There are three red-shaded weighting factors in the fan beam formula, which are not in the parallel beam formula. Specifically, capital R is the radius of a circular fan beam scanning trajectory, not needed for parallel beam scanning. Also, another variable, capital U, is involved, which is the projected distance between the X-ray source and a pixel to be reconstructed. If the scanning radius is sufficiently large, then U will be sufficiently large as well. Then the first two ratios will become 1. Hence, we have the parallel beam formula directly from the fan beam formula after the angular range of the fan beam scan is changed from 2 pi to pi. Now, let me ask you a follow-up question. Can you get the fan beam formula directly from the parallel beam formula? Let us look at this small green object in 2D, which is a 2D delta function. If you measure it with vertical X-rays on the detector array along the x-axis, then we have a 1D delta function after the projection. If we tilt the detector array by an angle alpha, the detector array along the x-axis will be along the x's axis. It can be readily proved that the recorded data will be a scaled-up delta function, with the scaling factor being the reciprocal of cosine alpha. Look at the left figure. For a black detector array that is perpendicular to the purple X-ray beam, the 2D green delta function will become a 1D delta function. Now the red line is your detector array in fan beam geometry, and the X-ray beam in purple goes through a small green object and hits the red detector array in an obtuse angle. As I explained on the previous slide, the 2D green delta function will become a magnified 1D delta function due to the obliqueness of the red detector array. Note that the angle between the black and red detector arrays is denoted as gamma. Geometrically, it is easy to see that the red-shaded weighting factor is the reciprocal of cosine gamma, which corrects the magnified projection value to the counterpart in the parallel beam projection. We further need to understand that a divergence beam projection, a fan beam projection in this case, magnifies a delta function on a 1D detector array, as shown on the left. This comment also applies in the case of a 2D detector array, as shown on the right. With a 1D detector array, the delta function is simply magnified by the geometric magnification factor. For an object irradiated with a divergence beam, if the object is small, the angle it spans is also small. This is called a weak perspective. The projected object can be well approximated as a parallel beam projection of the object after being scaled up by the magnification factor. In other words, the divergent beam projection of a small object can be regarded as a parallel beam projection subject to a scaling up operation. With that scaling up effect, you understand why you got this ratio RU. Clearly, this green dot will be magnified by this factor RU. Note that in the fan beam formula, this ratio has been squared, so we need dig deeper to explain the squaring operation. To find the reason for the second ratio factor, I mentioned that angular variables are different in fan beam and parallel beam geometries. The fan beam angle is denoted as beta, while the parallel beam angle is theta. As shown on the left, the beta de theta is U over R when the green object is close to the origin. Then we see that the ratio needs to be squared. The last weighting factor is 1/2. This one is easy to explain. Because the fan beam formula assumes a full fan beam scan over 360 degrees, it measures each line integral in the parallel beam formula twice. Hence, we must apply 1/2 to compensate for doubly sampling. With these geometric insights, we see that these three red-shaded weighting factors are all intuitive. From this observation, we can directly obtain the fan beam formula from the parallel beam formula without the derivation steps in the textbook. The heuristic explanation of the fan beam formula is not only interesting but also useful in extending the fan beam formula that was originally derived for a circular scan trajectory. Have you noticed that all the geometric arguments I used are individual view-based and don't assume a constant radius value? While the fan beam formula in the textbook assumes that R is constant based on my heuristic analysis, even if R depends on the fan beam angle, the same fan beam formula should still work. This idea was published in 1993 out of my PhD research. We did numerical simulation with various quite random scanning trajectories. Our extended fan beam formula consistently produced excellent reconstruction results. I know your group uses CNT-based detector arrays with non-circular scanning trajectories. Please stay assured that this non-circular fan beam formula works well in practice, although it is not theoretically exact. Early 1990s, I finished my PhD dissertation, which mainly solved two problems. One is the long object problem. In material sciences, many samples are elongated instead of being sphere-like. For X-ray microtomography, cone beam projection data are necessarily truncated along the longitudinal direction. At that time, micro-CT reconstruction required that a spheric sample be completely contained in a cone beam X-ray projection without any data truncation. To solve the long object problem, I derived a generalized Feldkamp algorithm for cone beam spiral CT image reconstruction. Later, we found that this had, and still have, major applications in medical imaging and homeland security screening. The other problem I solved in my dissertation research is the flat object problem, since many material samples are plate-like. To solve this problem, I proposed another adapted Feldkamp algorithm. Other planar samples are fossils, semiconductor chips, car batteries, and others. Recent years, this area becomes increasingly important. It seems coincident that both the long and flat object solutions have generated lasting impacts. How to do image reconstruction in the cone beam geometry. Again, I like to use a heuristic approach. In this case, the 2D fan beam is extended into the 3D cone beam, and we have two variables to index a location on the 2D detector plate, including an additional zeta variable. An X-ray from the source goes through an object and hits the detector plate at the red dot. The cone beam reconstruction formula in the blue box is very similar to the fan beam formula in the green box, but with a different weighting factor. How do you interpret the new weighting factor for cone beam CT reconstruction formula with the additional zeta in the denominator? It is not too difficult to justify the new weighting factor in blue color. In fact, the blue factor on the left-hand side is the product of the black factor and the green factor on the right-hand side. As you see, the ray of interest going through the object makes a vertical angle alpha and a horizontal angle beta. These two angles are needed to specify the narrow X-ray beam or X-ray line in the 3D space. As I argued before, we need to do the cosine correction for the angle beta. Then by the same token, we need to do the second cosine correction for the angle alpha. Putting these two cosine correction factors together, we arrive at the blue weighting factor. The similarity between the fan beam and cone beam formulas can be alternatively explained as follows. We can use the fan beam formula to do image reconstruction on every transverse slice if we have enough fan beam data on that slice at a longitudinal position. For a given green point in an object, we don't have a fan beam on the transverse slice to cover that green point, but we do have a tilted fan through that green point from a desirable orientation. That is, when you view from top to bottom, the tilted fan and the desirable fan overlap perfectly. Thus, we can use the red ray in the real cone beam to approximate the orange ray in the imaginary fan beam within the transverse slice in light blue after a cosine correction is applied for the angle alpha. Note that the cosine correction for the angle beta has already been incorporated in the fan beam formula. Although the cosine correction, also called obliqueness correction, is approximate in estimating the virtual horizontal fan beam data, the reconstruction results are often practically excellent. Part of the reason is that such a cosine correction is point-specific and thus quite adaptive. 2 decades ago, spiral CT was developed from fan beam geometry to cone beam geometry. Cone beam spiral CT has now become a dominating imaging mode. Again, there are about 300 million medical CT scans globally every year, with most of these scans performed in the cone beam spiral scan mode. Our work on cone beam spiral CT has been well recognized in the field, as remarked by peers. So far, what I have explained are the key ideas about approximate fan beam and cone beam CT reconstruction. Naturally, we want to find methods for exact cone beam reconstruction of a 3D object. As approximate cone beam reconstruction was developed based on the parallel beam formula, for exact cone beam reconstruction, we first consider 2D parallel beam projections recorded with a detector plate. This is a limiting case when the cone angle is very small. The simplest 2D parallel beam scanning method is to rotate the 2D X-ray parallel beam for 180 degrees around a fixed axis and decompose the 3D reconstruction problem into many 2D tasks to reconstruct parallel transverse slices defined by the green rings on the left. These tasks are not new, as we already know how to reconstruct a 2D cross-section from either parallel beam or fan beam projection data. In a 3D space, we are not restricted to rotate around only one axis. In fact, we can illuminate the object from any orientation as we wish, say along the normal vectors from any trajectory on the Mobius strip. Do we have a Fourier slice theorem in the 3D case? Based on the heuristics I used to justify the Fourier slice theorem in the 2D case, we can immediately have this 3D Fourier slice theorem. Look at a 2D parallel beam projection along the green vector. We should not be surprised that the 2DFT of this 2D red parallel beam projection will give you a 2D blue slice in the 3D Fourier space, and this blue slice will have the same direction as pointed by the same green vector. This Fourier domain slice reflects all the wave components of the original 3D red object that propagate in the directions orthogonal to the green vector. According to that 3D Fourier slice theorem, a 2D parallel beam projection along the green direction can be Fourier transformed into the spectrum on the plane orthogonal to the same green direction. For a 180-degree 2D parallel beam scan, the green vector can be rotated within K2-K3 plane from the positive K3 direction to the opposite direction. Clearly, this scan will cover the whole Fourier space K, 1K, 2K3 for the inverse Fourier transform. Now, an interesting question is, what if the parallel beam projection direction traces a 3D trajectory as shown on the right? An important observation is that as long as the starting position of the normal vector of the K1, K2 plane is along the K3 axis, and the end position of that vector is opposite to the K3 direction, no matter how the plane is rotated and wobbled, the K1, K2 plane will still cover the whole K1, K2, K3 space seamlessly. Let me convince you as follows. Look at the left part of the slide. Let us color the top surface of K1, K2 plane red and the bottom surface blue. After the initial normal vector along the K3 axis is turned upside down along any 3D trajectory, you will see the bottom blue surface on the K1, K2 plane when you view from top. If we color the points in the upper space red and the points in the lower space blue, any red point which initially saw the red surface now sees the blue surface. For this change to happen, relative to the double-colored plane, a red point must have gone through this rotating plane to the other side of the plane. In other words, this red point is measured during the tomographic scan. Similarly, any blue point is measured as well. This is the so-called Orlov theorem, implying that an Orlov scan allows exact image reconstruction through the inverse Fourier transform. Again, this consideration has assumed parallel beam projection geometry. Next, let us see how the Orlov theorem is instrumental for exact cone beam spiral CT image reconstruction. First, we need several elegant and critical concepts. A PI segment is a line segment with its endpoints on one helical turn. It can be proved that every point in a long object contained by a helical scanning trajectory, such as the blue ball in the left illustration, is on one and only one PI line. The helical trajectory between the two endpoints of the PI segment is called a PI arc. The TAM window is defined as the detector area on a cylindrical surface delimited by two consecutive helical turns, which is also called the TAM-Danielson window, on which we distribute X-ray detector elements densely. Clearly, the PI segment can be viewed as a diameter of a sphere, and the blue ball is illuminated from the PI arc and recorded on the TAM detector window. If the X-ray cone beam is a 2D parallel beam, the blue ball can be exactly reconstructed according to the Orlov theorem. Our key argument is that since the blue ball is very small, the cone beam angle is so small that we can use the weak perspective approximation to convert the cone beam data to parallel beam data after a proper scaling up or weighting step to reconstruct the blue ball as accurately as we need, which means that exact cone beam spiral CT image reconstruction is indeed feasible. Based on this heuristic consideration, the Katsevich cone beam reconstruction formula can be re-derived. To elaborate my claim on the previous slide further, I'd like to underline that the blue ball I mentioned can be at an arbitrary 3D location. Hence, any small ball or small target can be exactly reconstructed from cone beam spiral CT data. Since the Fourier slice theorem implies that image reconstruction is done through the inverse Fourier transform, which is a linear operation, the original object consisting of many small balls or targets can therefore be exactly reconstructed from spiral cone beam projection data according to the Orlov theorem if small balls on each pi line inside a volume of interest are covered by cone beam projections recorded in the TAM window. In contrast to exact reconstruction of an arbitrary small ball, an approximate cone beam reconstruction algorithm, such as the generalized Feldkamp algorithm, will be unable to reconstruct the ball exactly due to the approximate obliqueness correction involved. For example, a ball will be reconstructed into a star. Similarly, all the balls in the object will be reconstructed into many stars, and the superposition of these stars will give an approximate cone beam reconstruction only, since errors cannot be cancelled out perfectly during the superposition operation. The original papers on exact spiral cone beam image reconstruction are filled with complicated formulas and many derivation steps, which can be mathematically followed line by line, but the motivation behind these lines is hidden from imaging engineers and students. Hopefully, the essential idea behind all these mathematical details can now be heuristically appreciated based on what I have explained so far. This area of cone beam CT is very unique due to data truncation and beam divergence, and also very important for various practical needs. Here I mention our triple source cone beam spiral CT reconstruction papers in collaboration with Professor Katsevich and other colleagues. By now, I have basically covered the theory of analytic image reconstruction for cone beam spiral CT. Some of you asked me earlier to talk about AI imaging a little bit. Given the limited time, let me mention contemporary general AI, especially physics-inspired generative AI models such as the diffusion model. A representative image dataset is needed for the diffusion model to learn the distribution of all images to be reconstructed in an application domain. In the forward pass, every training image, say a green image on the left, is reduced slightly and then degraded by a tiny Gaussian noise at each step. After many such steps, the image eventually becomes a pure Gaussian random field. Then we can learn a score function from all the noisy data. After that, according to the learned score function, we can turn each random field back into a realistic image under the original data distribution. For example, put a pure noise image on the right in the bottom line, we can perform many small denoising steps until a pink image is finally emerged. Note that the noisy image on the right in the bottom row looks like a random field, but per request of the seminar host, it is actually a 2D barcode for students to scan using your iPhone as evidence that you attended this seminar. Let me magnify the barcode for your convenience. Please take a photo of this barcode now. Thank you. The diffusion model has demonstrated the state-of-the-art imaging performance, outperforming the famous generative adversary network in many tasks. To explain why the diffusion model can model a complicated data distribution so well, let me reveal the key heuristics on this slide. As we know, a continuous function can be well approximated as a piecewise linear function or a sequence of impulse functions. Similarly, a smooth data distribution can be well represented as a mixture of Gaussian distributions. In the training process of the diffusion model, we are essentially learning this Gaussian mixture where each small Gaussian noise is a component in the Gaussian mixture model. The power of generative AI can be best explained in the framework of statistical inference based on the Bayesian theorem. Many medical imaging problems are special cases of Bayesian inference. The Bayesian formula embodies the essence of rational decision-making under uncertainty, rigorously updating our belief based on new evidence. In the past, maximum a posteriori map image reconstruction has been the holy grail to reconstruct the optimal image that is most probable given the observed data and prior knowledge about underlying images. By maximizing the posterior probability, map reconstruction balances the respect to the observed data with constraints imposed by the prior knowledge. However, it is well known that the main challenge in achieving the map reconstruction has been the lack of a high-quality model for the distribution of images to be reconstructed. Now, modern generative AI solves this problem elegantly. With the diffusion model, we recently achieved promising results in few-view breast CT reconstruction. The clinical significance lies in the potential to transform breast CT from a diagnostic tool to a screening tool if we can just use 1/2 or even 1/3 of the number of cone beam projections for breast CT. We are also exploring the potential of generative AI-based stationary CT image reconstruction. This is our system reported in a recent IEEE journal for ultra-fast small animal micro-CT imaging with 29 X-ray source and detector pairs. If you are interested in more details on modern generative AI, I recommend our latest review article on this hot topic. In addition to generative AI, medical multimodal multitask foundation models are also a major direction for AI in medicine. If you are interested, you are welcome to watch my YouTube channel. For example, this is the seminar I gave recently for radiology department at Wisconsin University. Finally, I acknowledge my funding support from both NIH and leading companies. Also, I am grateful for my students, postdocs, and many world-class collaborators, and thank you so much for attention. Now, I am ready to take your questions, and if you want, you are welcome to email me as well.